Dear students, I again welcome you in the second lecture on the theory that is the Oparian Haldane theory. Now, in the first lecture, we discussed in the Oparian Haldane theory the first point, that's the formation of Earth and its primitive atmosphere. And in the first lecture, we lastly began to discuss about the second point, that is the chemical evolution of life uh, or the chemogeny. So we are discussing, this is the second lecture on the theory of this protobiogenesis or the Oparian Haldane theory. Now we shall be discussing the second point. The first point we have already discussed it in the first lecture. The second point in the theory of Oparian and Haldane is the uh, chemical evolution of life or chemogeny. So we say that in chemogeny, simple inorganic molecules which were present upon the primitive earth, like the methane, carbon monoxide, carbon dioxide, hydrogen, ammonia, etc., they reacted to form the complex inorganic molecules. And it is these complex inorganic molecules which later on aggregated to form the simple, simple organic molecules. Simple organic molecules. So I will again repeat, these are the simple inorganic molecules which are present upon the primitive earth. They reacted to form complex inorganic molecules. This is a molecular aggregation. Now there is question. For this molecular aggregation, we need energy. Where from the energy came? You will see upon the primitive earth there were some sources of energy like the UV radiations, cosmic radiations, and X-rays. During that time there was no ozone layer upon in the stratosphere. No ozone layer was formed, so the ultraviolet radiations directly came upon the earth. These are high energy radiations. With these radiations, cosmic rays and X-rays also came on this very planet. So this was one source of energy. Now the second source of energy was that there was continuous lightning, and due to the lightning electric discharge was created, which was another source of energy upon this planet. And uh, the last source of energy was that volcanic eruptions were continuously going on, and from these volcanic eruptions also energy was derived. So there were three main sources of energy. One is energy derived from the solar radiations like UV radiations, and the second is the energy derived from the lightning due to the electric discharge, and the third is energy derived from volcanic eruptions. All this energy was used over here for the process of molecular aggregation. The simple inorganic molecules aggregated into complex inorganic molecules, and the complex inorganic molecules aggregated into simple organic molecules. For example, the simple organic molecules are amino acids, simple sugars, nitrogenous bases, etc. And after that, the molecular aggregation went further one step more, that is, from simple organic molecules, complex, from simple organic molecules, complex organic molecules were formed. Complex organic molecules. These complex organic molecules are the first biomolecules, like the uh, what we call as RNA, like the proteins, like the glycerol, like the fatty acids, like fats, etc. And from these complex organic molecules, the first protocells were formed. The protocells, or we say from these complex organic molecules before protocells, some structures were formed which we call as conservates. Conservates. These conservates are sometimes also called as the first metabolic capsules of life, as they showed some of the properties of living organisms. For example, they showed division by budding, as is seen in some of the living organisms even today. So these are the first molecular aggregates which showed some signs of life. That's why sometimes we also call them as first metabolic capsules of life. These conservates then formed protocells, protocells, and it is these protocells which formed the first cells. And when the first cell was formed, the what we call as the time was 3.5 billion years ago. So this marked the beginning of life on this very planet. So we say that first, simple inorganic molecules aggregated into complex inorganic molecules, then complex inorganic molecules aggregate into simple organic molecules, which again aggregate into complex organic molecules, which gave rise to the first metabolic capsules of life on this planet, that is conservators. Or the conservators further developed and formed protocells, and from the protocells, the first cells were formed. But during this whole process, energy is required, and as this is the process of molecular aggregation, and I already discussed it, where from the energy came. Now, the second question is that all this is possible due to the absence of the free oxygen, what we call as the reducing atmosphere. All this molecular aggregation is possible due to the reducing atmosphere. Because if the reducing atmosphere might not have been present, in other words, if the free oxygen was present, this molecular aggregation might never ever have had happened. Because the free oxygen never allows this type of molecular aggregation. It degrades the larger molecules by the process of the oxidation. So, this molecular aggregation was possible due to the reducing atmosphere or due to the lack of the free oxygen on the primitive Earth. But one more point I will like to add over here is that once the molecular aggregation was going on, you will see once the molecular aggregation was going on, the conservates were formed. Conservates. These conservates are also known as the first metabolic capsules of life. Now, among these conservates, you will see many of the biomolecules were developed, like the RNA, like the RNA, like the proteins, and like the what we call as the fatty acids, glycerol, etc. Fatty acids, glycerol, simple sugars, etc. But over here, I would like to add one more point that the first information molecule which was formed on this planet among the living organisms, that was RNA. So RNA served as a first information molecule, and this period is called as RNA world by some of the geneticists and some of the molecular biologists, call this period when the whole information was present in the RNA, when there was no DNA. Some geneticists and molecular biologists prefer to call this period as RNA world. RNA world, because then there was no DNA. The RNA was the sole information molecule, or the RNA was the sole nucleic acid. And later, gradually, later on, what happened? The RNA molecules began to serve as biocatalysts. That is, they also began to serve as enzymes. So, besides being carrying out the function of information molecules or nucleic acids, they began to what we call as work as biocatalysts or enzymes. But later on, what happened? Once the DNA was formed, it proved as a more efficient information molecule as compared with the RNA. That is why the RNA was replaced as information molecule by DNA. That's why you are seeing in all the higher organisms, in all the higher cells, the information molecule is DNA. But the RNA was once information molecule in all the living organisms. It is indicated by the presence of RNA as genetic material even today in some of the viruses. And you will see after the DNA was formed, the proteins also begin to get formed, and these proteins invaded the area of biocatalysis. That is, they began to form the enzymes, and the RNA was replaced also from the biocatalysis field, and the biocatalysis was taken away by the proteins. As you see that the enzymes are the enzymes you will see are the biocatalysts. They are made up of proteins. But there are still in the present world, there are still indications which point towards the fact that RNA has been the biocatalyst in the primitive living organisms. There are indications, because you will see we even today say that all proteins are all enzymes are proteins except ribozymes. What are these ribozymes? Ribozymes are those enzymes which are made up of RNA. Although very few ribozymes are present, but they are even found today. For example, peptidyl transferase, ribonuclease P, etc. So even today, some of the enzymes which are found, they are RNA in nature. Although majority of the enzymes, 99% of the enzymes are made up of proteins. So still we say that all enzymes are proteins except ribozymes. So this presence of ribozymes even today, it indicates that sometimes in the ancient past, the RNA served as biocatalyst, because the proteins, once they evolved, they began to serve as better biocatalysts than the RNA. That's why RNA was replaced by the proteins in the field of biocatalysis. And DNA, once it evolved, it served as a better information molecule than RNA. That is why the RNA was replaced in the field of the this information molecule, or in the formation of nucleic acids by DNA. So DNA replaced RNA in terms of the information molecule, and proteins replaced RNA in terms of the what we call as biocatalysts. Now, still you will see this is an interesting thing, that once you study the central dogma, you will see it is DNA, which is the information molecule, and it is proteins which are ultimately formed in the central dogma, and this whole process goes through RNA. Though that type of RNA is messenger RNA, so we say that the information molecule present is DNA, and the products of these information molecules are proteins, but the whole process goes through the RNA. But there was once a time when the information molecules used to be from the RNA, and once the biocatalysts used to be from the RNA, as there are indications even today, which we discussed. So this is what we call as the point which we call as chemogeny. So I structured this point to make you explain what's RNA world, and the first information molecule we developed was RNA. So my idea over here was to discuss the chemogeny. So in chemogeny, we simply say that from simple inorganic molecules, complex inorganic molecules developed, from complex inorganic molecules, simple organic molecules developed, from simple organic molecules, complex organic molecules developed, from complex organic molecules, the conservates developed, then protocells, then cells. This is the process of molecular aggregation, which was possible due to the absence of life, and due to the presence of the due to the absence of the free oxygen, or due to the presence of the reducing atmosphere. So after that, there is the last point in the theory, that is the third point, that is the biological evolution of life. Biological evolution of life. This is the last point in the Oparian Haldane theory, that's biological evolution of life. In the biological evolution of life, we discuss about as to which were the first organisms which developed on this planet. So we say that the first living cells which developed on this planet were similar to the present day archibacteria, or to the present day mycoplasma or PPLO. And after that, you will see the first living organisms which developed, they were chemoheterotrophs. The first living organisms, with respect to their mode of nutrition, were chemoheterotrophs. It means that they fed upon the molecular aggregates which were formed on the primitive broth. They fed on these molecular aggregates. That's why they called as were called as this. They were called as chemoheterotrophs. But once the temperature began to decline, the molecular aggregates were formed on a slow speed now, and due to their continuous consumption by the chemoheterotrophs, there was considerable decrease in the number of these organic molecules present on the primitive broth. Now after that begin to develop the chemo autotrophs. Autotrophs. These chemoautotrophs. So the first mode of nutrition on this planet was chemoheterotrophism. Later on, the second mode of nutrition was chemoautotrophism. Now these chemoautotrophs began to convert the simple inorganic molecules present on the present in the primitive broth into the organic molecules. So these are the chemoautotrophs. So after that, from chemoautotrophs, the what we call as anaerobic anaerobic photoautotrophs develop. Photoautotrophs develop. So these were anoxygenic or anaerobic photoautotrophs. What they basically did, they used they used hydrogen as the hydrogen sulfide as the source of hydrogen, and they didn't use it water as the source of hydrogen. You will see this is the carbon dioxide, and they used H2S, means the hydrogen sulfide, as the source of the hydrogen, and as such they evolved what we call as they evolved they formed that is this glucose C6H12O6, and they formed what we call as the H2O, and sulfur was released. Okay, sorry, the hydrogen, sulfur as well as the hydrogen was released. So no oxygen was released in this process. That is, this is an oxygen. That is why we call it as an oxygenic process. We call it as an oxygenic process. The sulfur was released over here. So the chlorophyll began to develop, but there was no what we call as aerobic photoautotropism. Later on, aerobic photoautotropism began to develop. So first mode of nutrition was chemoheterotropism, then there was chemoautotropism, then there was anaerobic, there was anaerobic photoautotropism. Later on, developed the aerobic photo aerobic photoautotrophs developed. Now, aerobic photoautotrophs are which you see presently, plants are aerobic photoautotrophs. They simply do this is carbon dioxide. They use H2O as the source of hydrogen, and simply they form C6H12O6, and this is water and this is oxygen. Now the oxygen began to get evolved, and now the reducing atmosphere ended at this period, and this blocked the further evolution of the life as the molecular oxygen was now available. And after that, from them you will see the heterotrophs developed, the heterotrophs developed, and this is how the different modes of nutrition developed upon this planet. Now there is another concept you will see at this time when we are discussing the different modes of nutrition. All the life whatever existed, it was unicellular. Now from unicellular life, you will see from unicellular life, the colonial life began to arise. Colonial life means many unicellular organisms began to live in colonies, though this is not multicellular life, but this is definitely a move towards multicellular life. Now, from colonial life, the multicellular life began. The first cells which were formed, they are prokaryotic in nature, and from the prokaryotic cells, the eukaryotic cells were formed. It's a different. This is a different discussion. How from the prokaryotic cells, the eukaryotic cells are formed, as per the Lin-Margulis concept of endosymbiosis or endosymbiotic hypothesis. Many of the aerobic bacteria and many of the blue-green algae invaded the prokaryotic cells, and these aerobic bacteria later on developed into the mitochondria, that's a cell organelle present in the modern eukaryotic cells, and these blue-green algae developed into these chloroplasts, and this is how the first these plant cells were formed. In other cells, only aerobic bacteria invaded, and these developed into the modern eukaryotic cells. But there is contradiction of many biologists with this view. Some other biologists believe that due to the invagination of the plasma membrane or the plasma lemma, the cell organelles which are present inside the eukaryotic cells like the nucleus, Golgi, ER, lysosomes, etc., they developed. So it's a different discussion, but we need to remember over here that from simple unicellular life, the colonial life developed, which was a move towards the multicellularity, and the prokaryotes gradually, in the course of millions and billions of years, developed into the eukaryotes. This is all about the Oparian Haldane theory. So we need to remember that there are three main points in the Oparian Haldane theory. The first point is the formation of Earth and its primitive atmosphere. The second is the chemical evolution of life or chemogeny, and third is the biological evolution of the life.